Okay, so I've spent all day, which I love, like going through clients' mutations because I check every single one of those by hand because we don't really have a database that does a good job at predicting whether they're important or not. When it comes to genetic mutations, you can have a genetic mutation that, let's just compare it to a car, right? You can have a genetic mutation to where it looks like you got a fender bender in your car. It's gonna make your fender look funny, but your car's gonna run just fine. Maybe you have a mutation in the engine. That's a really big deal. You might just have like a busted tire. We can get you another tire. It's not a big deal. That's not a direct comparison, but it'll make sense if you know what I'm talking about. So I look at a bunch of different databases essentially to determine if that gene mutation is something that I should even like tell them about. Because I'm not going to tell them about something that's not pathogenic, and we have a lot of data to support that it's not pathogenic. So clinical variant database is a big one. It's called ClinVar. It's ran and managed by the NCBI or the NIH, so it's kind of like the government's one. And that one is kind of limited. So only about 10 to 30% of all pathogenic mutations, including ones that are gain of function, so not exactly pathogenic, but like different, right, are in that database. So to be in that database, you essentially have to have had someone look at you and say, let's get genetics to like follow up on this. And then when we look at people's genomics in that database, we tend to tag a bunch of things. So if they have this really weird condition, we'll kind of like just start tagging it. And sometimes it's because of that condition. Like sometimes it's directly causal, right? Or it contributory. But sometimes it's not. Sometimes it's just kind of like something weird that showed up. So it's not a causative database. None of these are. One of the best databases that I like to look at is the CAD database. I think it's by the University of Washington. So the CAD database combines like dozens of different annotations. So it looks at a bunch of different types of data and then uses machine learning to decide if that is a big deal or not. It will present what's called a Phred score, Phred, and from that Phred score, you can see if it's in like the top whatever percent of all mutations. So a Phred score of 10 or higher means that it's in the top 10% of all mutations. A Phred score of 20 or higher means that it's in the top 1% of all mutations. And a Phred score of, I think it's 30 or higher, is in the top 0.1% of all mutations. This is rank-based. It's not biology-based, so that doesn't mean that like something around a 10 couldn't be really bad. It just means that out of all of the available ones, it wasn't as bad as some of the other ones. The really good thing about this is it can handle coding versus non-coding. Some of the databases only look at coding. So if it's non-coding, if it's not something that converts into a protein later, it's not going to be in the database. But the CAD can handle that. It's also not mechanism specific. So sometimes your DNA contains this little sequence. It's called a translocation sequence. And your machinery in your cell has to bind to it and drag that enzyme somewhere else in the body. That is not great because enzymes sometimes have different functions depending on where they're located, and this won't tell you about that. It also won't tell you if the mutation is like in the catalytic site, so for like MTHFR, if it's where the folate goes, right? Or if it's in like the cofactor site, so for MTHFR, that would be a derivative of riboflavin or vitamin B2 called FAD. It doesn't tell us where it's at in that. It also doesn't tell you if it's in a regulatory site. So for the MTHFR example, the A1298C site is in the regulatory site. The catalytic site works just fine. It's just you can't turn the enzyme off when it's supposed to be turned off, so that's a problem. Also, we're training the model, right? It's a machine learning model. We're training it on data, so the data we give it could be bad. So it could have some training bias. So REVEL is also, REVEL is also another genomic variant effect predictor that you can use, and it's also a machine learning model. REVEL is going to combine the scores from other genomic variant effect predictor models like SIFT and Polyphen and MutePred and stuff like that. So with Revel, the scores go from 0 to 1, with 1 being that's bad, like that's definitely a bad thing, to 0 being like we're pretty sure that's not. Revel is only for coding, so it does not handle non-coding at all. The thresholds that I like for these, so the threshold I like for CAD when I'm looking at somebody's data to kind of like back up a little bit is 20. At 10, I start looking at the other numbers of the other predictions, like the prediction services, right? But at 20, I'm like, oh, okay, let's go ahead and find it in the protein. That's for CAD. For Revel, I start about around 0.5. I'm starting to be like, that makes me wonder. Let me look at the other ones. 0.7 is like pretty strongly pathogenic. And then 0.8 is where I go. I perk up and I'm like, oh, that's we've got a winner here, folks. Polyfin is one that's really used in like clinical genomics type services. And Polyfin is really cool because it predicts if the changing nucleotide, if that is going to correspond to a changed amino acid, right? If that new amino acid in the original amino acid spot, when we substitute that out, is that going to impact the overall structure and function of the protein? So, like, super cool. So for Polyfin, my kind of like lower limit, I start like looking really closely if it's 0.85. And Polyfin goes from 0 to 1 as well. The higher number is the most pathogenic. So 0.85 is the one where I perk up and I go, that, let me look at this again. If you go to GNOMAD, which is GNOMAD, so GNOMAD, I think, let me look. So it's GNOMAD. Just search that. If you type in the RSID in the top, so like RS7412 or whatever, and you scroll down, it's going to give you a list of like a CAD score of this, a Revel score of this, a SIFT score of this. So let me just tell you what my thresholds typically are, and you can kind of look through and check. So my threshold for CAD is 20. 20 is when I really perk up and start looking. Again, if it's a little bit below these, I'm going to look anyway, just because I'm curious and I'm nosy. But 20 for Revel, 0.7 is kind of when I start looking. 0.85 and up is when I'm like, oh, okay, yeah, that's important. SIFT is going to be reversed. So for all of these, the higher number means the most pathogenic, except for SIFT. SIFT is like the lower number is the most pathogenic. So for SIFT, my number is 0.05 and lower. That's when I start paying attention. Polyfin is 0.85 and up. Splice AI is 0.5 and up. Pangolin is 0.5 and up. And PhyloP is 4.0 and up. PhyloP is kind of weird. It's an evolutionary conservation score, so it essentially just means that like across evolution, when we look at all these different species that have this same enzyme, how conserved is the spot? Like how important is it that this spot stays what this spot is told to be, right? So you can kind of tell if you have, this gene that's like very, very important, and this one spot on it is very important to that function of that protein, it's gonna stay, right? So anything like above 4, I start paying attention to that just because like everybody has it in like that spot, so it's probably important. And Splice AI and Pangolin are deep learning models that are pretty good. Now they all have their strengths and they all have their weaknesses, so you have to be able to like look at all of the numbers and say, like it's, coming up that it's a problem in like 2 of these, let me go figure out why it's not in the other ones. I mean, it might just be that like that site isn't read. Like if it's non-coding, it's not going to be in some of these. And that's the same for everyone. All of them, most of them only read certain parts or certain mutations. But if you have an RSID and you want to know if it really is like a big deal or not, just go to GNOMAD or GNOMAD and put it in the search and it'll pop up all those numbers for you.